For decades, the field of cybernetics has operated on a strict assumption: noise is an error. To an engineer programming a rigid mechanism to navigate a physical space, structural precision requires eliminating random variables. The system must filter out the chaos to function accurately. But look closely at biological navigation at the cellular level, and you see a process defined by chaos. Organisms continuously tumble, drift, and eradicate through unpredictable fluid environments. They do not filter out the randomness, they rely on it. In biological systems, a random process is not an obstacle. It is the mandatory pool of variation required to generate a precise outcome. We can observe this computation across 3 distinct scales of life: the navigation of gut bacteria, the development of the insect brain, and synapses in the visual system. Understanding how these systems function forces us to rethink how life builds complex, sustaining architectures. Biology does not use deterministic blueprints, it computes with randomness. Consider the bacterium E. coli. It must navigate the human gut to find higher concentrations of nutrients, like sugar, but it lacks a sensory organ to see its environment. It has no map and no way to aim toward a target. Its physical hardware is highly restricted. The bacterium is propelled by a molecular wheel attached to a flagellum, a corkscrew-shaped hair. It has no steering mechanism. It can only toggle between 2 possible motor states. This diagram outlines those 2 mechanical states. When the molecular wheel spins counterclockwise, the flagella align and propel the bacterium straight forward in what is called a run. While this run state provides forward momentum, it occurs in a strictly random direction. The bacterium has no physical capacity to align this forward vector with the actual location of the sugar. If the wheel reverses to clockwise rotation, the flagella push outward against the water. The forward momentum breaks, causing the entire bacterium to randomly tumble in place, reorienting it blindly for its next run. Neither mechanical state allows for intentional aiming. Every straight run is arbitrary, and every tumble resets the orientation to a new, equally arbitrary angle. The movement is entirely blind. To solve this navigational deficit, the bacterium utilizes a single-step memory constraint. It computes by comparing the current local sugar concentration to the concentration it experienced a moment prior. It applies a strict probabilistic rule to this data. If the sugar concentration is increasing, the bacterium increases the probability that the motor stays in the counterclockwise run state. It simply biases the system to keep moving in whatever random direction it happens to be facing. If the concentration is decreasing, the rule flips. The bacterium increases the probability of the motor switching to clockwise rotation. This forces a random tumble, breaking a mathematically unproductive path. The individual bacterium never learns the actual location of the sugar source. It simply samples its immediate surroundings, looping this simple probability algorithm over and over. We can observe the macro result of this process in the bottom circular plot. The jagged, erratic vectors show how a population of bacteria, through many individual biased runs, eventually aggregates around the area of highest sugar concentration. This algorithmic process is known as a biased random walk. By probabilistically favoring positive outcomes over negative ones, a population of organisms uses blind, erratic noise to reliably locate and stay near a target. This utilization of noise extends beyond spatial navigation. We see the exact same computational principles applied to the physical construction of neural architecture. In the Drosophila fruit fly, researchers observed an anatomical paradox. The brain is mirror-symmetric, meaning the left and right sides share identical genetic instructions. Yet specific neuron populations vary unpredictably between the 2 hemispheres, producing anywhere from 15 to 45 cells. This variation is the result of notch signaling. It's a developmental process called lateral inhibition, where neighboring cells engage in a competitive, algorithmic game to suppress one another's growth. Initially, cells are locked in a balanced feedback loop. Each secretes a protein telling its neighbor to stop growing, while receiving the exact same suppression signal in return. Then, symmetry breaks. Random developmental noise tips the balance. One cell gains a slight advantage, creating a winner-takes-all cascade, completely suppressing its neighbors. The structural rule of lateral inhibition is absolute. Exactly one cell will win the space. But because the symmetry breaking relies on random fluctuation, the specific identity of the winning cell is completely unpredictable. Biology programs this noise into the sequence as a bet hedging strategy. By generating varied physical traits from the same DNA, the species produces a wider distribution of behavior, ensuring enough diverse traits survive unpredictable environmental shifts. The fly's visual system presents a cybernetic sorting challenge, routing nearly 5,000 individual photoreceptor axons into 800 exact synaptic groupings, or pixels, in the brain. The top half of this neural superposition diagram charts the optical layout of the insect's eye. Dotted lines track how 6 physically distinct lenses all capture light bouncing off the same point in visual space. The solid lines descending into the brain show the structural solution. Those 6 disparate signals are routed down and grouped precisely into a single unified neural cluster. The system achieves this through pre-specification. Algorithmic growth pathways act as physical funnels, forcing the correct developing axons into the correct general anatomical neighborhood. But once inside that local neighborhood, the mechanism changes. The neurons do not connect using a strict deterministic lock-and-key code. Instead, they exhibit synaptic promiscuity. These neurons are programmed to make connections at all costs. If the right partner isn't found, they connect with the wrong partner. Proving growth relies on probabilistic connections, rather than absolute molecular blueprints. Relying on rough physical pre-specification and highly promiscuous synapses leaves a network messy. In mammalian visual systems, like the developing ferret retina, the brain must actively prune and fine-tune these loose connections to achieve visual acuity. This tuning process occurs before birth. The retinal network must finalize its wiring with ever receiving light, meaning it has zero environmental input to guide calibration. To substitute for missing environmental data, genetics deploy a mathematical trick. The retina spontaneously generates random sweeping waves of electrical activity across the field of neurons. Crucially, the wave directions are entirely random. By crossing the tissue from orthogonal angles over many iterations, they ensure only physically adjacent neurons consistently fire together. This mechanism utilizes a core biological principle, known as Hebbian pruning. Cells that fire together wire together. If neurons fire at different times, their connections weaken and dissolve. This is post-specification. Because the waves or waves are in direction, only neighboring cells correlate their activity, allowing the system to weed out the messy outliers and slowly achieve the absolute matrix precision required for functional high-acuity vision. The classic equation of genotype plus environment, creating an individual phenotype, is incomplete. Simple gut bacteria, insect neural asymmetry, and the pruning waves of the mammalian retina all share a common mechanical reality. They achieve highly specific goals without relying on rigid deterministic encoding. The genetic code explicitly encodes stochastic processes to solve logistical bottlenecks in space and time. This is why even identical twins possess unique fingerprints and differing neural maps. That variance is not strictly a byproduct of environmental nurture or genetic mutation, but programmed developmental noise. Biological self-organization embraces this noise because the flexibility of probability is more robust than a rigid system. If the blueprint is too brittle, the system cannot adapt to environmental change. In the algorithms of life, noise provides the fundamental raw material required to build precision.